Hey there guys, it's Mr. Herbst here. I'm going to go ahead and do a little walkthrough of our study guide, which is going to be due before our genetics test. My goal is to keep this video under about 15 minutes, so I'm hoping that you all can set aside that short amount of time to watch it. The genetics test is going to be one of the biggest tests that you guys are going to have all semester. Anyway, let's go ahead and begin. This first chart right here, at first glance, may be a little bit confusing, but what it's asking us is to compare and contrast meiosis versus mitosis. Now, I'm going to go ahead and start with our drawings here. In mitosis, let's recall here that we start with a cell, and how much DNA is in that cell? Well, let's go ahead and say 100%. All the DNA in your body is in that one cell. And when that cell goes through mitosis, prophase, metaphase, anaphase, telophase are all represented in these arrows. It splits into two daughter cells, each with 100%. So each of these two daughter cells here are identical to the parent cell. So the products of mitosis are they're sort of like clones. They're sort of identical to one another. And let's go ahead and answer this question. Identical to parent? Yes. How many cells are produced? Well, the answer is 2. Now, in meiosis, something a little bit different happens, where we start with a cell that's 100%, and we go ahead and we do, just like in mitosis, we go through prophase, metaphase, anaphase, and telophase, but we actually go through it twice. And we end up with, actually, this time with 4 cells, and each of these 4 cells is really weird. Each of them has 50% of our DNA. And if you recall, each one of these things is a certain type of cell called a gamete. So meiosis is a process that produces gametes. And some examples of gametes, almost guarantee that you heard of these at one time in your life. Sperm and egg. Those are what we call sex cells, and those are used in sexual reproduction. If we didn't go through this process of meiosis, we would end up having individuals that would begin to look very, very weird in just a few generations. So it's a good thing that we do this. How many cells are produced? The answer is 4. Are they identical to parents? Well, you take a look. Each one of these has 50%. Is that the same as the original parent cell? The answer is no. Down here, guys, you'll notice this. Although doing something like this is really valuable, I'm going to go ahead and cross this out. And I'm going to do something a little bit different, and I'm going to go ahead and show you guys kind of how sexual reproduction works. Well, as I've just discussed, we start with a male, and we start with a cell that has 100% of the DNA. And that cell is going to have to go through a process called meiosis, and it's going to produce 4 cells, each that has 50% of our DNA. And as I previously discussed, what type of gamete is this? Well, these are sperm cells. So I'll indicate that with some little tails here. And then females, they start out with a cell that has 100% of the DNA. And they go through meiosis. And meiosis produces 4 cells that each have 50% of the DNA. And what's kind of interesting that happens with females is that these three cells right here, they kind of donate all their stuff to just one cell, and they die off. So as I discussed, what gamete is women? Well, gametes produce eggs. And since these 3 little things right here, they're called polar bodies, donate all their stuff to this one cell right here, that cell gets real big. Actually, an egg cell is one of the only human cells that you can see with your naked eye. It's a very large cell. Then, these males and females, they go through a process called sex. And the sperm over here fertilizes the egg. And when 50 and 50 come together, what do you think we get? That's right, we get 100%. And that is the original cell that you and me all started out as called a zygote. Anyway, a lot of these terms here are not going to be super important for you guys to know for your test. I just kind of wanted to make sure we understand kind of how sexual reproduction works. I'm going to go ahead and make it a little bit more specific. Let's say that this is a male human being. How many chromosomes are in a male human being? Well, the answer is 46. And so how much would be in each one of these gametes if they all have 50%? Well, the answer is 23 chromosomes. So they all have half of the total number that you're supposed to have. Same thing with females. They start with 46, and each one of these here has 23. And then when males and females go through the process of sex, 23 and 23 come together, and then we end up with 46. And that's a good thing. That's what creates you and me. Anyway, guys, go on to the next page. These terms right here, I'm going to go ahead and represent these here with pictures. Homologous chromosomes, we can say that well, we got to kind of remember, what does that term homo mean? And homo means same. So in a sense, they're kind of like the same chromosomes. So if I had a chromosome here and a chromosome here, these chromosomes here are kind of like they're chromosomes for the same type of trait. So for example, if we're talking about eye color, as we've discussed, you have two alleles that contribute to your eye color. Maybe this chromosome over here gives you the big B, and this chromosome over here gives you the little b. So they're homologous in the sense that they both go ahead and contribute to your eye color. A tetrad is just the pairing up of those homologous chromosomes. So they kind of like come together in prophase 1. And crossing over is as a biologist, I think it's awesome what crossing over is. It's sort of like where the two chromosomes sort of, they come together and they sort of lock and link together. And then synapsis is where they sort of like share a little bit of that DNA that is right in here. So after these guys cross over and they split back apart, they're going to look like this. Do you see how kind of some of that DNA got swapped over? That occurs in prophase 1 of metaphase, of a meiosis. And what is the purpose of this? I mean, it's kind of difficult to understand sort of actually how important that is. But I'm going to try to visualize that with a couple of visuals. Let's say that I have a couple of chromosomes here. We kind of know that some chromosomes are bigger than one another. So in this case, I have chromosomes that have noodles, and these chromosomes have mastacholi. Well, when these chromosomes here split, we're going to have mastacholi go from here to here. But is there anything kind of new that's being split? Well, the answer is no. What about when these guys split? Is there anything kind of new that ends up being split? Is there anything kind of different? Both of these bowls right here still represent the same thing that they kind of started with. But what if we go through that process of crossing over? What if we kind of do this, where the chromosomes kind of share? So if these two bowls represent chromosomes, what if they kind of shared some of each other's DNA and sort of mixed everything up here? Now, when they go ahead and split, we kind of have like this whole jumbling mess of different kinds of differentiation going on. The correct word is genetic diversity. Because these chromosomes here split up, it allows for our human race to kind of be, so everybody can kind of be different. And right down here, guys, 5 and 6, although these are really important terms, we are not going to be focusing them on this unit. So you can go ahead and skip that part right there. My definition of complete dominance would be that one allele always masks another. And to put that in different terms, it's where like we don't have both alleles show up at the same time. One or the other shows up. And so we've talked about this in class where we can have brown is typically dominant to blue or something like that. Incomplete dominance is where we have a blending or a mixing of alleles. So for example, if somebody was big R, and that stood for red, and somebody was little r, or a flower was little r, and that stood for white, well, if they had one of each, they kind of, these two colors here blend together and kind of make pink. So some human examples of that would be like skin color. For example, President Obama is a great example of how skin color sort of kind of mixes together, where his father was black and his mother was white, and he sort of became, when they got together, he was a little bit lighter than his father. So the skin sort of mixed together. Codominance is where we have both alleles show up at the same time. So for example, we can have big N, and that might be normal. And little n, or big S might mean that somebody has sickle cell anemia. And what if they had big N and big S? They have both normal and sickle cell blood showing up at the same time. So it's kind of where they have like mild anemia. And that's due to the fact that they have both normal red blood cells, and then they have these sickly looking ones showing up at the same time. So a great example of where that's found in humans is sickle cell anemia. Multiple alleles is where we have several alleles that contribute to the same trait. And we've actually talked a lot about this in class, where we discussed blood types, where we can have allele A, which corresponds with blood type A, allele B, which corresponds with blood type B, and allele O, which corresponds with blood type O. And sex-linked traits, as I just discussed on the last page, those are traits on the X chromosome. And so what we kind of do when we find traits on the X chromosome is we write a gigantic X, and then maybe a little subscript N would mean normal. And then what if we had a little n right there? That would correspond with colorblindness. And a good example of a human trait that is sex-linked is colorblindness. For number 9, if we were to show that black fur color is dominant to white in mice, just let's make a key for that. Well, big B would mean black, little b would mean white. For 10, write the genotypes for each of the following using the key from above. Well, if somebody was homozygous dominant, we kind of have to remember what that word homo means, and that means same, and dominant means that they have the capital letter. So somebody with a mouse that was homozygous dominant would be big B, big B. Heterozygote would be big B, little b, because the word hetero means other, so otherwise known as one of each. And homozygous recessive would mean 2 of the little alleles. On to the next page here, guys. The next problem here is a little bit more complicated, but it's stating that we have 2 red flowers that cross. I'm going to go ahead and do part B first. It's asking us to first identify the specific type of genetic relationship that we see. And it's not actually giving us very much information other than telling us that we have these numbers. And if we look here, if we take 20 and sort of divide it into each one of these, we find that it's kind of like we have one red flower for every 2 pink ones for every 1 white one. So we have a relationship of 1 to 2 to 1. And more specifically, where we have red and white, but then we also have pink. That should be a buzzword. It's sort of a mixing of red and white. This is a good example of incomplete dominance. And right here, we see that we have 51 red flowers for every 21 white ones. Well, if we take 21 and kind of divide it into 58, I know the math doesn't work out correctly, but it's kind of like we have 3 red ones for every 1 white one. And we don't find any pink ones at all. So what could be the possibilities of the parents that would give rise to these offspring? Well, we kind of have to work backwards and sort of think about it a little bit. But if we find white ones, we know for sure that if we have a white one, that white one would have to be little r. No other choice, because little r is for white and big R is for red. And if we go ahead and work backwards, what would have to be up here in order to have there be a little r down there? Well, the answer is little r right there. That would also mean that little r would have to be in this box and this box. But then it also says that we find 3 red ones for every 1 white one. So that means that these other 3 boxes all have to be red. The only way to do that is if they were big R. So in this box here we have big R, big R, big R, little r, and big R, little r. So in this case, the parents of these offspring are big R, little r. And in this case here, we're going to go ahead and work backwards again. Since we know that one of them has to be white, 1 out of the 4 is white, and there's no other choice for it to be but little r, little r. What would have to go up here in order for a little r to go there? Well, it would have to be a little r. That would also mean that little r would have to be here. And then we find that 2 of them have to be pink. In order to be pink, what do you have to be? You have to be big R, little r. So what would have to go in here in order for these guys to be pink? Well, no other choice but to be the big R. And so that would mean that there would have to be a big R here in order to put a big R there. And there would have to be a big R at the top here to put a big R there. So in this case, our cross is once again big R, little r crossing with big R, little r. And 14, describe the ABO blood system by writing out all the keys. Well, as we've stated in class, we can have type A blood. We can have type B blood. We can have type AB blood. And we can have type O blood. Now, what's all the possibilities for somebody to be type O? As we've discussed, O is a recessive allele. So the only way somebody can be type O is to have 2 of the O's. And how about for type A? The only way somebody can have type AB blood is to have AB alleles. Now B, it's possible that somebody could be big B, but since the O is recessive, it's possible that somebody could be big B, big O. And with A, for it to make type A blood, it's possible for somebody to be big A, but it's also possible to be that. Number 15 asks us to show the cross between a type AB man and an O woman. So the first thing that we do is write down what type of genetic relationship this is. And since it's dealing with blood type, I've explained in class that should be a buzzword, multiple alleles. 2, write out our whole key. Well, what's all the possibilities for somebody to be type A? Well, they can be big A, or AO. How about type B? They can be BB or BO. Type AB, the only choice is to be AB, and type O, the only choice is to be OO. And then for 3, state our cross. Well, it's saying that we have an AB man. Well, our AB man, the only choice is to be AB. We'll have an AB crossing with an O woman. The only possible choice for the woman genotype is OO. We'll go ahead and do our Punnett square here. I like to put the women along the top and the men along the side. And then 5, answer the question. Show the probabilities of genotypes. We'll start with that. We only find 2 possible genotypes within this Punnett square. We have AO and we have BO. And the probabilities of somebody being AO is 50%. The probability of somebody being BO is 50%. And then with phenotypes, we only find 2 phenotypes in here. We find type A blood and we find type B blood. And the probability of somebody being type A blood is 50%. The top probability of somebody being B blood is another 50%. And 19 here, guys, seems pretty complicated, but it's asking us to create our own sex-linked pedigree for colorblindness. Now, in class, we discussed that colorblindness is recessive. So what I kind of like to do is say that when we have the big X, big N, that's for a normal individual, and X little n is for somebody that's colorblind. And now I'm going to go ahead and give you guys a pedigree here that we're going to sort of figure out on our own. But let's say that we have a female and a male, and the male is colorblind. When we shade in circles or squares, it represents that that is a person that's affected by the certain recessive disorder. In this case, it is colorblindness. And then those individuals there have 3 baby girls. Girls are once again circles. This woman here married a normal man, and they had 2 kids, 1 female and 1 male. And this male was born with colorblindness. One of their daughters right here was born with colorblindness. And then this daughter here married a man. And those 2 individuals there got together and made 1 male or 1 man that had colorblindness, 1 normal female, and also another normal baby daughter. Now, the first thing that it wants us to do is label the generations. Well, up here is generation 1. Down here is generation 2, and this is generation 3. Now, the easiest thing to do when we're solving one of these Punnett squares is to start with the males and figure out, look at the shaded in males and figure out their genotype. Because all of these males have something in common. They all have to have a Y, meaning that they only have one of these possible X's as their genotype. In this case, because they are all affected by colorblindness, they have no other choice but to be X little n. All those males are X little n. And this female right here, because she is a female, she has 2 X's, but she has colorblindness. So that means she has to have both of the little n's. So now, let's figure out kind of what is it that this man gave to this woman that Now let's kind of work backwards. If this woman here was got her X from this man right here, where did she get the other X that gave her colorblindness? Well, she must have gotten it from mom. So mom must have at least 1 little n. But since her square is not filled in, she is normal. She must have at least 1 big N, otherwise known as being a carrier. These individuals here, once again, these 2 females, what did they get from dad? They got at least 1 little n. How do I know? If you think about it like this, what would happen if dad gave the Y away to these girls? Well, they would have been born men, and they wouldn't have been girls. What do they get from mom? Well, in this case, they would have had to get the X big N. How do I know? If they got the little n, their circle would have been shaded in. These men right here, these 2 men right here, since they are both unaffected and they all have something in common, they all have a Y, their genotype is no other choice but to be X big N. And so in this case, what is the genotype of this female? Well, if we look at what did this female here get from dad that made her a female? Well, she got this X big N. What did she get from mom? Well, if you look at it, she could have gotten the big N from mom to be normal, or she could have gotten the little n from mom and still been normal. Same thing with all these three girls over here. They all could be X big N, X little n. Anyway, guys, I realize I ran over my 15-minute time limit, but if you think about it like this, if you watched this video in its entirety and you learned one new thing, that was one question that you would have gotten wrong on the test tomorrow if you had not watched this video. So I appreciate the effort in class. You guys are doing a great job overall. Anyway, I'm signing off, folks. You all have a nice night.